Okay, we'll go ahead and get started on lecture 2. Probably one of my most favorite lectures. We are going to learn about the ABO blood group system. What makes you the blood type you are, okay? So like we mentioned in lecture 1, the ABO blood group was discovered by Carl Landsteiner in 1901. He actually received the Nobel Peace Prize for his work in this, but not until I think it was like 1932 that he received that award. So the ABO blood group is the most important blood group system. So we're going to start adding on blood group systems as we go further into this course. So ABO is the most important. And why is that? Because our ABO antibodies are of IgM classification for the most part, and they can cause immediate cell lysis. And if you give somebody the wrong ABO, that is fatal, okay?
Why Peace Prize?
Nobel Peace Prize?
Why not chemistry?
I do not know why. I don't know. So there's 4 major phenotypes. We have group A, group B, AB, and O. So what that means, if you ask yourself when you're trying to determine what type of patient is, group A patients have the A antigen on their red cells. We type our patients by what antigen is present, okay? So group A have the A antigen on their red cells. B have the B antigen on their red cells. What do you think about AB? It has both the A and the B antigen being expressed on their red cells. What about the O? Neither, okay? So really, group O is not an antigen, okay? It's not related to an antigen. It really is the lack of both the A and the B antigen. Does that make sense to everybody? Okay, so remember that. Carl Landsteiner's rule states, and I will say this several times in this course, the antigen that you lack, you can build the antibody to, okay? So if type A patients have the A antigen on their red cells, they lack the B antigen. If we were to test for ABO antibodies in your patient plasma, what antibody, ABO, is going to be in your patient plasma? Anti-B, okay? They're type A because they have the A antigen on their red cells, but they have anti-B in their plasma. Okay, Carl Landsteiner's rule states you can build the antibody to the antigen that you are lacking. Okay, so B has the B antigen on the red cell. He will have the anti-A in his plasma. What do you think about AB? They don't have either ABO antibody in their plasma. Okay, they have both the A and the B antigen on their red cells. No ABO antibody in their plasma. Okay. What about O? Anti-A and anti-B. Your group O people actually even build a separate antibody. It is A comma B. It is not a combination of anti-A and anti-B. It's its own molecular structure. Okay? It's only produced by group O, and it is of, everybody listening to me? It is of IgG classification. All of the other ones are IgM. What is significant about anti-A comma B being of IgG class? It can cross the placenta. So when we start talking about moms and babies and we have hemolytic disease of the newborn due to ABO incompatibility, that means mom is type O, baby is some other type A or B. Okay, so that means mom's anti-A comma B is coating baby cells causing hemolytic disease of the newborn. I say that now, I know that's kind of beyond what we're talking about right now, but I don't want to confuse you when we get to moms and babies and I start telling you our ABO antibodies can cross the placenta. Only one has the capability, and it's only when mom is group O. Okay, so if mom is O, baby is either A or B, that A comma B can cross the placenta and coat baby cells. Okay, I tell you that now because you're going to think that all of your ABO antibodies are IgM. True to an extent, except for that A comma B produced by group O. Okay, does that make sense to everybody?
So does that mean AB patients can get whatever blood products from whichever?
Ah, good question. So your A and B patient has the A antigen and the B antigen. They don't have any ABO antibodies in their plasma, correct? So that's the concern when we're transfusing red cells. We want those red cells to be compatible. We want to make sure that your patient's antibodies aren't going to attack the donor red cells. So because AB patients don't have any of the ABO antibodies, they are what we call the universal recipient. They can get A, they can get B, they can get AB, and they can get O. Okay? Versus, take a look at O. He has neither the A antigen or the B antigen, has anti-A and anti-B in his plasma. So if he were to get A cells or B cells, what's going to happen? His antibodies are going to attack the donor units. Okay, that would be an example of wrong ABO given, and that would be intravascular hemolysis. Okay. Versus A, they can receive A or O. Okay, O is our universal donor. Okay, he has no A or B antigens on the red cells, so he can go to any of these guys. However, he can only receive O red cells. Okay. Question, Aaron.
Is there any difference in susceptibility to infectious diseases among these individuals because of their lack of presence of antibodies in the blood? Because it said we're forming these antibodies in response to the environment.
I just read something the other day about that. Let me pull that article up. There's certain infections that have been linked to the type of ABO. I just read an article about that the other day. So I'll come back to that question. So your ABO is determined, it's genetic, we all know that, and it's actually a codominant gene from chromosome 9, okay? So you'll get one gene from your parent, from each parent. And we'll talk about the Punnett square in just a little bit. All right, so that's kind of just what I said. This kind of demonstrates what we talked about. The antigen, each of these little dots represents the antigen on the cell. So for AB patients, notice that they have both the A antigen and the B antigen, no ABO antibodies in their plasma. Okay. For type A patients, they only have the A antigen, so they have anti-B in their plasma. B has the B antigen, so they have anti-A in their plasma. And then your group O people, they don't have any of the ABO antigens on their red cells, so they will have both A and B in their plasma. Okay.
Where does the positive and negative?
So positive or negative refers to the Rh or your rhesus factor. So in this course, you guys notice as we go through builds on itself. So we start out with just the forward and reverse type. Okay, what antigen is on the red cells? That's your forward typing. Okay, so when somebody says what's your forward type, what antigen is on your patient red cell? Your reverse type, what antibodies are in your plasma? Okay, that's your reverse. So forward and reverse. So ABO is just your forward and reverse typing. In lab number 3, we'll add on the Rh. And in lecture 5, I believe, we'll start talking about the Rh antibody system. But there's a misconception there too. So Rh really has a connotation to be associated with the D antigen. But as we start talking about families of antigens, they run in families. They have very similar characteristics, reaction phases, temperature. The Rh family actually includes D, big C, big E, and little e. That's your Rh family. So to say somebody's Rh positive, it's understood that they're D positive, but there are also other red cell antigens in the Rh family. So just keep that in mind. So all of your antibodies are going to be grouped in families based on how they react and what their characteristics are. So we'll get there too. Okay, but does everybody understand that your patient blood type is determined by what antigen is on the red cells? And you will have the opposing antibody in your plasma. Okay, I want to make sure everybody understands that. You have to understand this moving forward in blood bank. Okay, and you have to remember Carl Landsteiner's rule. If you lack the antigen, you can build the antibody. And that's why I want you guys to think about blood bank forward and backwards. Okay. And then we mentioned this, group O is an amorph. There's actually not an O antigen. It's just termed group O because they lack both the A and the B antigen on their cells. Okay, they don't have either of those antigens. And so remember, all people, according to Landsteiner's rule, can build the antibody to the antigen they lack. So for a group B person, they have the B antigen, they can build anti-A in their plasma. For a group O, has neither A or the B antigen on their red cells, so they will build both anti-A and anti-B in their plasma. And then group AB has both antigens, so they will neither have anti-A or B. So we talked a little bit about this in lecture 1. Remember that your red cell membrane is composed mostly of phospholipids and proteins, and it's the glycophorin is the major glycoprotein that attaches to your ABO antigens. If you think about your ABO antigens, they're a carbohydrate extending off of your protein there. So here's your protein, you have your glycoprotein, and there's your carbohydrate. This is where your ABO antigen is located. Your what determines your ABO? So you have these carbohydrates, actually oligosaccharide, so it's composed of 4. And then we're going to talk about a specific gene that will add a sugar, and then depending on if you have expression of the A or the B antigen, we'll add on an additional sugar, and that determines your blood type. Okay.
And the O's just have the base sugar.
What's that?
The O's just have the base sugar. They don't have the additional.
So we're going to talk about what happens. So the ABO system is actually controlled by another gene too. It's the H gene, and it's also I think it's chromosome 19. And so what the H chromosome does, if you are, whether you're homozygous or heterozygous for H, it is going to add an additional sugar. It's going to add L-fucose to this oligosaccharide. Okay. What's going to happen to that L-fucose, if you are group O, you lack the A and the B antigen. So no other sugar, and I have a drawing that I'm going to draw this out for you guys. There's no other sugar added onto that oligosaccharide. Okay. And so you just have that straight oligosaccharide in group O. So you'll have L-fucose being exposed. Okay. If you're group A or group B, there's 2 sugars that are going to be added. If you're group A and adds a sugar, it masks that L-fucose. All right, so it masks the amount of H. We're going to talk about that in an upcoming slide. So if you take a look at this cell membrane, and you have these carbohydrates extending throughout the cell, all right, so you have your ABO antigens dispersed throughout that cell membrane. Okay, so it's not just a couple. ABO are very antigenic, they're very prominent. And when we start doing our blood bank testing and we start grading our reactions, our ABO reactions are typically very strong, 4+ or 3+. And this is why, because there are plenty of antigen sites spread across the cell membrane. Okay? So you also see ABO antigens not only on your red cells, that's primarily where we focus on them in blood bank, but they can also appear on lymphocytes, on platelets, most of your endothelial cells, and some organs as well, such as kidneys. And you could even have ABO expressed in body fluids. But primarily in blood bank, we are testing peripheral blood. We also later in the semester will get into HLA typing and determination of ABO RH for organ transplants. We'll start talking about that too. Okay, and that's why in organ transplant sometimes ABO is very important. Maybe not so much in bone marrow transplant. So if you guys ever known somebody that had a bone marrow transplant, it might have been of a different ABO. What happens in those patients is their type starts changing. So that if it's an A patient that receives a B bone marrow transplant, they will start producing B cells. So it's kind of interesting, and we'll get there. That's an advanced topic.
Would the antibodies start attaching to those?
So if you think about a bone marrow transplant, you think about the stem cells, they're going to start producing cells from the recipient. So those lymphocytes that are stimulated into plasma cells, and then your antibodies, your patients should start building the donor antibodies as well. So what's going to happen is you might have some initial hemolysis, but once the donor cells take over, then your donor cells will be present, and it should not, the recipient antibodies should not attack those cells. So you can actually detect ABO in early as 5 weeks in utero. So once that baby is 5 weeks old, they have their own ABO type. And that ABO type can be different from mom, okay? And we're gonna talk about the Punnett square in just a little bit. There's actually fewer antigen sites on the newborn cells than there is adult. They're there, but they're just fewer. But full expression of those ABO antigens should occur 2 to 4 years. And then they'll start building those ABO antibodies about 3 to 6 months. So here's the frequency of your different types of ABO based on the US population according to what race you are. So the majority of the white population is ONA, okay? And then there's some other ethnicities as well. Okay, so that just kind of breaks down. Keep this chart in your mind. We're going to start talking about frequencies, calculating what percent of the population it would be O positive and little c negative or something like that. So you'll need these frequencies. If on an exam, I don't want you to spend time memorizing these frequencies, okay? If on an exam I give you a frequency question, I will give you your ABO percentages. Okay, there's other topics in blood bank that you need to memorize, not so much on this, okay? Now you'll start remembering, however, 85% of the population is Rh positive, 15% of the population is Rh negative. You will need to know that, okay? But we'll get to that. All right, so here's some various genotypes that determine your phenotype. So if you are type A, you can either be homozygous, meaning that both alleles are expressing the A antigen, or you could be heterozygous, meaning that one allele is of the A antigen and then the other one doesn't have the A or the B being expressed. Okay, so you could be AO. For the type B, you could be homozygous with both alleles having the B antigen expressed, or you could be heterozygous, meaning only one allele has the B antigen. For AB, one allele is A, one allele is B. Okay, so you have sugar being added to the oligosaccharide for A and B. Okay. For group O, you have to be homozygous. Neither A or B is being expressed on either allele. Okay, does that make sense to everybody? So some students get confused and want to say that AO is a type O person. No, you have to be homozygous. Okay, you cannot have any expression of the A or B antigen in order to be group O. Okay, so we talked a little bit about this. I kind of touched the surface. The A and the B antigen formation, how do we detect that? It is dependent upon the H-antigen, and the H-antigen is inherited independently of your ABO, okay? And it's genetically controlled by a different gene. It's called the H gene. So you're either homozygous for H, meaning both alleles are expressing H. You're either heterozygous, meaning one of those alleles is expressing H and the other one is not, or you're homozygous, meaning that neither of those alleles are expressing or producing any of the H substance. Okay, what that means, this is very, very rare. If you don't have any of the H substance, do you think you have to have, what the H substance does is it's going to add the L-fucose sugar to the oligosaccharide. If H is absent, if you are producing no H, then L-fucose cannot be added. Therefore, regardless of what ABO you are, what antigen you have on the alleles, we would not be able to detect that in our blood bank testing. Okay, this is a rare blood type. It's called Bombay type. It's very, very rare. And if you guys know anybody with Bombay, if they are ever in an accident and need a transfusion, very difficult to find compatible blood for them. Oftentimes, if you are diagnosed with Bombay, you know you're Bombay, you are storing your blood. Probably every 5 or 10 years, you're donating the blood and you are paying to have it stored. It's that big of a rarity. Okay, so that's called Bombay type. We'll talk about it a little bit more extensively in a little bit. Okay, so what that does is the H antigen is going to add the L-fucose to the oligosaccharide. Once that L-fucose is added, your group A allele or your group B allele, whichever one you are, is going to add a further sugar. It is going to mask that L-fucose. Okay, that builds our oligosaccharide. That determines what type you are. And I'm going to draw this out in the schematic, and it's going to make perfect sense, I promise. So the ABO system is dependent upon the H gene expression as well, okay? So your oligosaccharide, like we mentioned, you think about that red cell membrane, it has to be attached, that oligosaccharide has to be attached to some type of carrier molecule, right? Typically, it's our red cell membrane. That oligosaccharide composed of 4 sugar molecules linked in a simple linear form, or it can even be in a branched form. So I want to go ahead and draw this out to you guys so you guys see it. And this B1 for, or beta 1 for, beta 1 3, that has to do with the binding. You guys don't have to go that deep, okay? All I want you to know is what sugars are going to be added depending upon if you're A or B, okay? Or both. Okay. So remember, your oligosaccharide chain has to be attached to a carrier protein. That's usually your red cell membrane. Okay, so here's your red cell membrane. Remember, our ABOs usually extend from a carbohydrate from that cell membrane. Okay, so here we go. This is the molecular level of that carbohydrate. Okay. Remember, your oligosaccharide is a combination of 4 sugars. All right, so you're going to have your galactose, and excuse my drawing. You'll have N-acetyl glucosamine, and this is in your notes. It should all be there as well. Okay, this is another sugar. And then you'll have another galactose. And like I said, you don't have to worry about the exact binding site. We're not going to get that technical. Okay, if you have the H substance, okay, so you're either homozygous H, heterozygous, meaning that only one allele is producing the H substance, or you're Bombay, meaning that you have no production of H. If you are Bombay, okay, this is what your oligosaccharides look like. It can never go any further than this. Okay, so if there's a chart at the end of lecture 3, I believe, 3 or 4, has all the different reactions for blood bank and ABO determinations. If you look at Bombay, he looks like he's an O patient, because what's going to happen is for patients that are group group A, they are going to add an additional N-acetyl glucosamine sugar on here. Our reagents detect that sugar, okay? So if no additional sugars are being added to this oligosaccharide, then our testing is going to think that they're O, even though the patient might actually have the A and the B gene. We do not know without doing a molecular genetic level test. We cannot test with our serological techniques in blood bank. Okay, does that make sense to everybody? So this is what Bombay looks like. They are going to look like a group O patient. That being said, remember Landsteiner's rule. You can build the antibody to what you lack. Notice that our patient does not have any H being produced. What does that cause? They're going to have anti-H, okay? So your group O patients that have the H sugar, the L-fucose added, our screening cells are all group O. So this patient's anti-A is going to react with our screening cells. If you look at your chart, they have a positive screen. 4+ probably on all 3 or 2 or 3 cells, depending on what type of screen you're doing. Okay, so that's usually how we're going to detect that this patient, they're going to react with all of our panel screening cells, because all of our panel screening cells are group O. Okay, so that is Bombay. Does that make sense to everybody? They are producing no H substance, so that L-fucose cannot be added at all. Now, irregardless, they might be AA, they might be AO, they might be BO, might be DB, they could even be AB. With our serological testing, we don't know. We cannot tell. Okay? It'll have to be a molecular phenotype done. Okay, so that's Bombay. All right, so let's say you're group O. Group O. You're having H substance being produced, okay? H substance is going to add an another sugar that's glucose. That's what H does. That's your type O right there. Because there is no A antigen or B antigen to add an additional sugar, okay? So your type O, if you are ever asked what group has the most amount of H substance, it's group O, okay? That is a really good board of certification exam question. They might ask you that. Because the ABO system is determined, H substance has to be there for the addition of L-fucose. Okay. All right, so that's O. Does everybody understand that? All right, if you're group A, we're adding on the A antigen is going to add on an additional sugar. And the location, I'm going to add it right here, actually. And it is going to be in acetyl galactosamine. Galactosamine. That sugar makes your patient A. Okay? So when we do our blood bank testing, we add our reagent anti-A. For the molecular structure of that, we're looking for this sugar on the oligosaccharide, on the carbohydrate attached to that red cell membrane. Okay? Does that make sense to everybody? That's group A. Should have left that there. Group B adds on another sugar, right? Remember, fucose is there, right? Fucose is there. We still have our H substance being produced. Fucose is there. The sugar associated with type B is D-galactose. That's your type B. Okay? So H substance being produced, H adds on that L-fucose, then the B antigen is going to add on that D-galactose sugar. Okay? That is what determines a B patient. They have that D-galactose sugar on the oligosaccharide. What do you do about an AB patient? They have both sugars being added to the oligosaccharide, okay? So they have D-galactose and N-acetylgalactosamine being added.
Do they both attach to the same galactose?
It adds, let me see the exact location. There's this one right here in your lecture notes is the one the schematic I like the best. So for an AB patient, this one doesn't really give the exact location though. I feel kind of silly that I don't know your answer. I'm not exactly sure where the location is, but I will check. I will come back to you guys on that.
If it's just like
It's somewhere like
some of the oligosaccharides are B and some are A.
I don't know.
So, or if it's,
But so it'll be on this oligosaccharide. So this is one oligosaccharide, so it will have both sugars being expressed. But I don't know if it comes off the lactose or the glucose. I don't know exactly where. Good question. Please stop me. Does that make sense to everybody? What sugars? You guys need to know what sugars associated with each antigen. Okay. And the importance of H.
there's actually the slide 19 indicates that there's some A saccharides and some B.
Oh, okay.
So it's a mix, but slides, and just slide 19.
Oh, wrong button. So let me see. Oh, that makes sense. You know what? I think I knew that. I did know that, but
We all.
put me on the spot, you know? Yeah. So Aaron's right. When that comes up, I'll show you. That makes total sense now.
So AB, see how like some of the oligosaccharides will have N-acetylgalactosamine, some will have D-galactose added on it. That makes it AB. So both sugars are being added. The thing you need to remember about AB is you have both the A antigen and the B acting on the H. Your AB groups are going to have the least amount of H substance because they have both A and B acting. And once those either N-acetylgalactosamine or D-galactose is added, it masks your L-fucose, which is your H substance. So if we were to measure the amount of H substance, our A1B patients have the least amount of H substance. Okay.
And that's just what I said there. Those A and B substances are going to mask your H. In the group O, remember, no further sugar is added, so that oligosaccharide stops at the L-fucose, which means you have high amounts of H. Okay, so group O has the highest amount of H substance. And we talked about Bombay. So what I want you guys to know about type O is there's no O antigen. I want you guys to understand that. It's termed group O because they lack both the A and the B antigen. Okay. And group O has the most amount of H. And here's a schematic. It's a good thing for you guys to know this based on the amount of H. Okay, so remember that we said, oh, there's no additional A or B antigen to add on a additional sugar. So N-acetylgalactosamine or D-galactose is being added. So we have high amounts of O. And then we haven't talked about their subgroups of A yet. So if you say a patient is type A, it's really understood that they would be type A1. Okay, there are hundreds of subgroups of A. It would take us all semester probably to study all the subgroups of A. Okay, so for this course, if we say the subgroup of A, they're going to be A2. And we're going to talk about the difference between A1 and A2. It has to do with the structure of the A. So A2 is not as strong as A1. So therefore, it is not as well at masking all of the L-fucose. So that's why A2 will have more H substance. Okay, followed by B and then A2B. Remember, when you have both antigens involved, you're going to have lower amounts of H being detectable. Okay, and A2 is again not as strong as A1, so that's why A1B is at the lowest amount of H because you have both N-acetylgalactosamine and D-galactose acting on that fucose. Okay, and then A1. That makes sense to everybody? This is the amount of H being produced. So O is going to have the most. A2B, A2B, A1, A1B is going to have the least amount of H because both sugars are being added to that oligosaccharide chain. And then here's our schematic. And again, remember, anyone that lacks the A or the B antigens, they can build the antibodies. Those antibodies are naturally occurring, okay? So you're going to build those antibodies from the air you breathe, the food you eat. They're naturally occurring, okay? And remember, they are all IgM association except for anti-A, B produced by group O patients. Now, when we are testing our patients for ABO, that is a room temp initial spin test. So we're adding our red cells in our reagent, we're adding patient plasma in our reagent cells. We immediately spin and read for agglutination. Okay, that's a room temp initial spin test. That's the term initial spin, okay? Because, guess what? All of these are IgM, cold reacting or room temp reacting antibodies, okay? They don't need that 37 incubation phase. One thing you guys have to remember about these ABO antibodies, it can bind complement. That's what makes a incompatible ABO transfusion so serious. It's immediate. Complement is going to be activated, resulting in cell lysis, okay? So an example, if you have a group O patient, okay, group O can only receive O red cells. You give them group A, your recipient's anti-A is going to attack the group A donor cells. That complement is going to be activated. Those donor cells are going to be destroyed. That's going to result in intravascular hemolysis. Okay, that's what happens in a wrong ABO transfusion. I mean, it's documented that cell lysis, cell destruction will occur at 1 mil a minute. That's how fast this is happening. Okay. And what happens is the cells, complement is activated, cell lysis, cell fragmentation. Those fragments are going to get stuck in the kidney. Also, you're going to have those, think about the formation of the heme. You're going to have those globin dimers being broken down. They can also get trapped in the kidney. The patient's going to go into kidney failure, DIC. That's what makes it so fatal. And it happens fast. Okay. Your patient's going to let you know right away if it's the wrong ABO transfusion because they're going to start having immediate pain. They're going to start breaking out in a fever. They might start sweating. They're going to go into tachycardia, trouble breathing. Okay, that's what's going to happen. Not to scare you. Remember, I said you have to have a healthy fear. So as we start building in blood bank, I will tell you to double and triple check yourselves. Labeling, very important. Ensuring proper patient identification. You guys will become anal about accepting patient specimens. They have to be properly labeled. They have to be signed by the phlebotomist. Patient identification is huge because if I get Peyton's sample in and it was really Jenny's blood, sorry, Jenny is O and Peyton is A, I just killed a patient. So that's how serious it is.
Will that reaction be as serious with autoantibodies?
Not as serious with autoantibodies. The problem with autoantibodies is in blood bank, we can never get any negative reactions. So we're unable to rule out any allo antibodies and finding compatible units of blood. So for patients with autoantibodies, we can try to do what we call, and we'll talk about this, it's called a warm autoantibody absorption. We can try to absorb out that patient's antibody, but oftentimes you can do 3 or 4 treatments and it still won't work. That antibody is still going to be there reacting. So for patients with warm autos, whether it be drug-induced, whether it be a true autoimmune disorder in the patient, typically they're going to have incompatible cross matches. We give them least incompatible, like the least reaction in their cross matches that we can. So your desired would be like a 1+ reaction. So, but sometimes, I mean, I've seen warm autos react at 3+. So, and if you guys don't know blood bank reactions yet, the strongest is 4+, meaning that you have one solid cell button. Okay, that's a 4+, and then it goes down from there. If that cell button breaks up, 3+, 2+, 1+, then negative. So warm autos is blood bank headache. Yeah. There's a new drug that UAMS is using now, especially with it's with multiple myeloma patients. It's actually an anti-IgG drug, and it's called, I don't know the long name for it, but it's called DARA, and it causes pan-reactivity on these patients. So everything is positive. So your IAT, which is your antibody screen, it's going to be all positive. Your panel is going to be positive. Your DAT is going to be positive. Your elution is going to be positive. You can try to do a warm absorption. It's not going to help. And so identifying any underlying allobodies and finding compatible units is near impossible for these patients. So that's what blood bank is. That's the most current blood bank topic right now, is dealing with these patients that are on Darintumab, I think is what it's called.
What's the drug?
For those patients with multiple myeloma, trying to prevent the rapid proliferation of the abnormal plasma cell clones. Okay, we talked about group O producing anti-A, B. Remember, that is a separate antibody. Some students want to say it's a combination of anti-A and B. That's not correct. It has its own molecular structure. Okay, so it's only produced by group O. I mentioned a while ago some subgroups of A and B. What this is, it represents a variant gene at the ABO locus. And so what's going to happen is it produces the lesser amount of the antigen. That's why A2 is not as strong as A1, and that's why A2 has more H substance, okay? We'll focus more on A subgroups. There are some rare subgroups of B, but again, they're more very rare. In lecture 3, I think we'll talk about some, but we really want to focus on the subgroups of A. That's most commonly what you will find in, that's what you will encounter in blood bank. So remember Landsteiner's rule. What you lack, you can build an antibody to, okay? So the thing about these A2 patients, or another subgroup of A, there's AE, AX, A3, and there's hundreds of subgroups of A. Their molecular structure of the A antigen is different. It's not as complex as the A1 antigen. So they lack some of the A1 antigen expression. Therefore, they could build an antibody to the A1, okay? So that's where we get our subgroups. If you have an A2 patient, they could potentially have an anti-A1, and that will cause a typing discrepancy that we'll talk about a little bit later on. So of your people that are type A1, or of your group A population, okay, 80% of them are going to be A1, okay? The majority are straightforward A, no problems. 20% of that group is going to be A2 or a subgroup of A. That's when our typing discrepancies can occur. Okay. It's 1 to 8% of A2 people will build that anti-A1 that I was mentioning. If you're A2B, I think it's like 22 to 35% will build anti-A1. Okay. And why can they build anti-A1? Here's your molecular structure. Take a look. Anti-A1 phenotype, see how it has the branched form of the A structure and the linear form. Okay, this is your A1. This is why the A is so strong versus your subgroup of A or your A2 phenotype only has the linear. And so remember Landsteiner's rule, what you lack, you can build the antibody to. So for A2 phenotypes who are exposed to another A1 donor, they can build an antibody to this linear structure, okay? So A2 people can have anti-A1. Okay? A2 people can have anti-A1. Because A1 types have both the branched structure and the linear structure, it is impossible for A1 patients to have anti-A2. Does that make sense? Okay, so A2 can have anti-A1, A1 cannot have anti-A2. That often confuses patients. Okay? So A2 can build anti-A1. They can. I'm not saying they have to. I'm saying they can. Okay? About 8% of A2 people will build anti-A1. Okay? Does that make sense to everybody?
Is that just a matrix?
It is not as severe. It's still something that blood bank wants to be aware of. It is not as severe. Because your A2 phenotype, they have some of the A structure. They're just lacking the branch. So while they can recognize some A, they won't recognize all of it. So it's not as severe. But it could still cause a transfusion reaction. Maybe just not as severe as a completely incompatible unit. A couple things that we think about when we think about clinically significant in blood bank. Number one, is it going to cause a transfusion reaction? Number two, can it cause hemolytic disease of the newborn? And so we'll talk about being clinically significant in blood bank a lot. Some of our room temp reactive antibodies are more of a nuisance because you think about it, the human body is not at room temp, and we're usually at 37 degrees. So any of our antibodies that react at room temp but go away after 37 incubation, they're considered nuisance. Typically, blood bank will kind of overlook them. Now, if they react at 37 or beyond, they're considered clinically significant at that point. Okay, so we'll talk a lot about that throughout this course, clinically significant.
And like I mentioned, there's hundreds of A subgroups that are out there. But the main thing you have to remember about these A subgroups is they produce less, okay, so there's not as much A being produced, and then they're missing some of the A structure. That's what I was going to say. So there's less A antigen, all right, so they're producing less, more of that N-acetylgalactosamine sugar added, okay? Or does it say more? Less. So if you have a subgroup of A, they're less antigenic than A1, so you're going to have more H substance. That's right. Yeah. Another thing is because you have decreased A antigen, when we do our forward and reverse typing on these patients, remember I told you that our ABO reactions are typically very strong, 4+ to 3+? With these patients, because there's less A antigen, your reactions are going to be very weak, could even be negative, okay? That plays a very important role in donor testing, because what's going to happen is for these A patients, we should expect a 4+ reaction with anti-A, right? If it's weak or negative, it could be typed as what? Could be typed as a group O. What happens if a type A unit is labeled as an O? What type of patient is that unit going to go to? O patient. So you have a really A unit going to an O patient. That is a hemolytic transfusion reaction. Okay. So that's why identifying the subgroups of A are so important in blood bank, especially in donor testing. And we'll talk about this in a future lecture. You will see, if you have a subgroup of A that has built an A1 antibody, it will cause a discrepancy, meaning, when we say discrepancy, meaning that your forward type does not match your back type. So what you're going to see in these group A patients is a weaker reaction with your anti-A. You're going to have what plasma reaction do you expect with type A? You expect a reaction, they're going to have anti-B, right? So they're going to react with our B cells in the back type. They're also going to have a reaction with our A cells, okay? So you can imagine if your front type looks like an O and your back type looks like an O, that's why it's so important for donor testing that these subgroups of A be identified. We do not want a real A patient going to a O donor, okay? So we're going to see what that looks like in a future lecture. All right, so remember, antigens are located on your red cells. The antibody is in your plasma. And when we start doing our testing, when we do our forward type, we are using patient red cells with known commercial antisera. So our antigen is on patient red cells. We are providing the antibody. The antibody is known. So we know what the antibody is. If we have agglutination, that means the that antigen is present on the patient cells, okay? For the reverse type, we use patient plasma where the antibody is with commercial provided red cells. So we know the antigen, we're testing for the antibody. So forward typing, we're going to take patient cells mixed with commercial antisera. Right, that commercial antisera is your antibody, commercially manufactured. And we're going to talk about how that's manufactured in a future lecture. So in your forward typing, you're going to have, right now, 3 tubes. We'll build on that. You'll actually have more when we add in the RH. You'll have anti-A, anti-B, and anti-A, B. Don't be surprised if when you start your internships or you start working in a blood bank lab, they do not use testing with anti-A, B. Sometimes anti-A, B is only used to confirm subgroups of A.
okay? So what anti-A, B is going to do, if the patient has the A antigen on their red cells and the B antigen on their red cells, it will react, okay? It's going to react if your patient is A or B. It's going to detect the A antigen or the B antigen, okay? So if this is really handy, if you have a weak subgroup with A, where your anti-A may be very weak, or it might even be negative, there's a chance that your anti-A, B might pick it up. So then you would have a reaction with anti-A, B, letting you know that your patient is not O. Okay? O will have a negative reaction with A, B. If you have agglutination, I want to stress this. If you have agglutination, that means the antigen or the antibody is present. Okay. So here's what your reagents look like. They are color-coded to help prevent the adding of the wrong reagent. So your anti-A will always be blue. Okay. And this has commercially manufactured antibody A. Okay. Your anti-B is always going to be yellow. And it has anti-B, and then your A, B is clear. All right. So you're going to take your 2 drops of antisera, remember, because we always add a little bit more antibody than antigen. 2 drops of antisera, 1 drop of patient cells. Because we're forward typing, we want to know what antigen is on your patient red cells. Okay. And then here is an example. Remember I said a 4+ reaction where you have a complete whole cell button. This is what it looks like. Okay, versus a negative, where there's no cell button, no small agglutinates of cells at all. This is a negative reaction. That's the forward type. We're looking at the antigen on your patient red cells. The reverse type, we are seeing what ABO antibody is in patient plasma. So we're going to take patient plasma, we're going to mix it with commercially manufactured red cells. So you have A cells and B cells, A1 cells. A1 cells and B cells that you will run on every patient in blood bank. Forward and reverse typing. You guys ever heard of type and screen? Maybe you've heard that on medical shows. You need type and screen stat. That's what it is. So ABO RH is your type, forward and reverse, and then your screen is seeing if they have any antibodies. Your screen is your IAC, which we'll get to. Okay, so that's your type and screen. Your reverse, patient plasma mixed with commercial A and B cells. If your patient has the ABO antibody in their plasma, they will react with the cells. For instance, if your patient has anti-A in their plasma, you will have a reaction with A cells. Okay? If your patient has anti-B in their plasma, you will react with the B cells. So here's what your reverse typing cells look like. You have A1 cells and B cells. These are actual donor red cells that the manufacturing, the commercial facility, the vendor collected. They process. The antigenic profile is known. So we know these are A cells. We know these are B cells. Okay. And then when we mix with patient plasma, we can look for agglutination. So here's your grading agglutination. 4+, you have 1 solid aggregate, all right, of red cells. 3+, you have several large aggregates. 2+, you have medium-sized aggregates with a clear background, okay? The difference between 2+ and 1+. 1+, the aggregates are small. You usually have a very turgid or reddish background. So your aggregates should be a little bit smaller than a 2+, and also you should have more of a reddish tinge background, okay? Now, I have a disclaimer. Sometimes you might see a weak plus reaction. That means it's weaker than 1+, but it's not negative. I don't like weak plus. To me, you either call it 1+ or negative, okay? But you might see some reactions graded as weak plus. I never use weak plus. I don't like it. It should be 1+ or negative, okay? But to me, because look, tiny aggregate, turgid, reddish background.
as you can imagine, this already has subjectivity involved in it. Yeah. So how do you differentiate between tiny and small? All right. That's why I was like, don't use weak. If you have tiny, small aggregates, 1+ solves the problem. Okay. So you can imagine differentiating a 2+ from a 3+, differentiating a 1+ from a 2+. There's some subjectivity there. Okay. So on grading reactions, I am not too critical. As long as you don't call 4+ a negative, as long as you don't call, 1+ a negative, as long as you can tell me that it's positive, please don't, maybe not a 4+ or 1+. That's kind of, if you have a bunch of small aggregates there, definitely not a 4+, but anywhere from that 3+, 2+, 1+, I'm usually pretty lenient in the grading on that, okay? Because there's definitely subjectivity there. And when we get further into blood banks, sometimes all of our ABO testing that we've talked about so far is done macroscopically. You only detect agglutination with your eye. Do not ever look at ABO agglutination under the microscope, okay? However, there are some testing that we will do, especially when you add AHG, you have to confirm if it's negative with your eye, you have to look at the microscope and see if there's any microscopic agglutination. If you notice microscopic agglutination, it is indicated by the plus sign with a circle around it. That indicates that it was microscopically positive only. But for the first 2 labs, we will only be doing 1 through 4 and negative. Okay. And here's pictures of what they look like. So negative reaction, notice that there's no agglutination at all in your -2. Notice in a 1+ reaction, see how small those aggregates are? And you see how you kind of have a turbid background versus the 2+ reaction kind of has more of a clear background. Okay, that's kind of how you differentiate a 1+ from a 2+. Okay, and then a 3+, notice that your aggregates are a little bit larger. It's even sometimes very common that you'll see 1 large aggregate and several small aggregates. I'd still call that a 3+. Okay, and then a 4+ is 1 solid cell button. Any questions on grading? Again, you guys will get plenty of experience with this, and you'll feel comfortable. All right, so for your ABO typing, you have your forward typing, you have your A, your anti-A, your anti-B, your anti-A, comma B. A1 lectin is added on if you suspect any type of discrepancy. So if your forward and back type don't match, that's what we call a discrepancy. That is when you would add in your A1 lectin. Anytime you do A1 lectin, and we'll talk about what A1 lectin is when we talk about our discrepancies. Anytime you add A1 lectin, it has to be run in conjunction with A2 cells. Okay. And then your reverse typing is A1 cells and B cells. All right, so this should be in your lecture manual. These are some of the expected reactions for all of our ABO types. Notice, so anti-A, anti-B, anti-A, comma B, that's your forward typing, okay? So notice for a group A patient, you have a 4+ reaction with anti-A because the A antigen is on their red cells. You also have a 4+ reaction with A, comma B, okay? Negative with anti-B. So remember Landsteiner's rule, whatever antigen you are lacking, you can build the antibody to. So in your reverse typing, if your patient is A, your A cells are negative because anti-A is not there because the patient has the A antigen on their red cells. But you have a 3+ with your B cells because the patient's anti-B is reacting with the B cells. Okay, does that make sense? Versus, let's look at, notice patient B, okay, so his type, he has the B antigen on his red cells, he reacted with A, comma B as well, all right? And notice that he has anti-A in his plasma, so we have a reaction with our A1 cells. And take a look at your A1B patient, notice that he has 4+ reaction, anti-A, anti-B, and anti-A, comma B. Notice that your AB patient is negative with A cells and B cells because he doesn't have any of the ABO antibodies in his plasma. If your
results are given here for A1 lectin. So if you notice your A1 patient has a 4+ reaction with A1, A1 lectin detects the A1 antigen. And what if this patient was A2? If this patient was A2, we would have a negative reaction with A1 lectin. They've seen here in this example here, A2. So notice A1 and A2, the only difference in the reactions from A1 and A2, well, you have a little bit of a weaker reaction here, remember, because there's less antigen being expressed. So we have a weaker antigen with anti-A, and also negative reaction with A1 lectin. So that lets us know our patient is not A1. Okay. Does that make sense to everybody? All right, let's do some practice. All right, so here's your first patient. 4+ reaction with anti-B, 4+ reaction with anti-A, anti-B, and anti-A, B. What's your front type? AB. What do you expect your reverse reaction to be? Negative with both A cells and B cells. So that's what this patient is. So this patient is AB. Does that make sense? Notice how I asked you what your front type was, and then I asked you what your reverse type was. When we start identifying discrepancies, that's what you have to ask yourself. If I'm front typing as an A, but back typing as an O, that doesn't make sense. We have to investigate why that is. Is it a subgroup of A? Okay, that's kind of our hint that we're dealing with a discrepancy. When you're forward and back do not match. Here's another one, 4+ reaction with anti-A, negative with anti-B, 4+ with A, B, and in the back type, we have negative with A1 cells, 4+ with B cells. That's a type A patient. Do you know if that patient is A1 or A2? No. Without A1 lectin and A2 cells, you cannot tell an A1 patient from A2. Okay, so if you have a straightforward A patient, this is all you do, there's no discrepancies, no anything, we call our patient A. Okay? Most of the time in blood bank, we are not going to spend the time, tech time, reagent waste, and typing all of these A patients. Okay, if it's straightforward, forward and reverse match, they're just group A. Okay? Now, if you have a discrepancy, you had a reaction right here, meaning your front type and back type didn't match, then we would have to run A1 lectin and A2 cells to see what subgroup of A that patient is. But on a routine basis, they're just A. That's all. Most of the time, you're not going to have any problems, but it can happen. Okay, here's one, negative, anti-A, anti-B, and A, B, 4+ reaction with A cells and B cells. Do your forward reactions match your back reactions? And what type is your patient? That's type O. What if you had a 4+ reaction with anti-B and a 4+ reaction with your A cells? That's a type B. All right, those are really straightforward. You guys feel pretty good about those? Thing to remember is you type your patient on what antigen is present. Okay, so don't get confused on what your back type is. Okay, whatever your front type is. So if your patient reacts with anti-B only, then they're B. Okay, and then the back type is going to be the opposite because you can build the antibodies to what you're lacking. So they'll have anti-A in their plasma. So these are pretty easy. Do you think it gets more complicated? Oh yeah. And I finished right on time. Me too, Sarah. Isn't this fun? This like excites me. I guess I'm a nerd.
really started.
Position of the BloodBank type. Yes, so I talked to Kirk. Yeah,
And I think I know who he is.
Friday,
Yeah.
and he told me that the position is officially posted
Okay.
and that you guys can apply for it. So those of you that indicated interest, I will forward that link to you guys.
Okay. So, and he emailed me like 2 weeks ago after like you posted that,
Yeah.
and I emailed him. So I can't find it. Where is it?
Oh, the blood bank?
Now, I couldn't
Is
find
it on
it.
campus?
It's, yeah, just go to the careers. It's there now. It wasn't, but I just found it. But my email isn't registering. Like you have to log in to apply, and it says my email doesn't like it.
When did you try?
Today.
Okay, I'm wondering if something is wrong because I can't log into Blackboard.
Yeah,
Just
well,
like not I was able to access my email though.
Well, I can get into my email itself, but like when I logged into Blackboard, I couldn't use my email. I had to use like my number barcode at uams.edu, but it says my email username doesn't like exist or something.
That's weird.
And it's a problem.
I wonder if something's going on today.
Yeah, I've had that problem since like Friday and through the weekend. So I'm trying to.
Yeah. So did you find it on Blackboard? The careers through Blackboard?
No,
or
he
campus?
told me how to do it, but then I found it. If you go to the UAMS website and then you click jobs, it'll take
It was
you
on
to
there?
here, and then I just typed it in and it popped up.
Okay.
Because it wasn't.
Yeah,
it's
because
now.
I was like looking for.
Yeah, me too. I was like, I can't find it. You're welcome. That's what actually going to ask you if maybe